From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on August 28th, 2025. Hello, Micronauts, and welcome once again to our Quality Quorum. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 106th episode of Matters Microbial. Many thanks to every listener, viewer, guest, and supporter. Please keep those likes, listens, and views coming. First, I want to remind listeners and viewers of the recent loss of Dr. Moselio Schechter, a very influential figure in microbiology, and my friend. On his birthday a few years ago, my wife, Dr. Jennifer Quinn, put together this wonderful assembly of LuxArt images showing Elio at different ages and some microbial celebration. He was truly a microbial hero who touched so many lives, including my own. I honestly have never traveled that much, but I have been to the Netherlands twice, which is relevant to today's discussion. The city of Leiden, seen here, is beautiful. And Leiden University, seen here, is also lovely. Nearby in Amsterdam is Micropia, the world's only museum devoted to the microbial. You can see my happiness to visit that museum again here. And within that museum are several huge sculptures of my favorite myofauna, the tardigrade. My wife, Dr. Jennifer Quinn, had a chance to cuddle up to one, seen here. Next week is the beginning of the last course I am teaching here at the University of Puget Sound before retirement. Eek! Here you can enjoy this final class's logo, created by the great Dr. Liza van der Aert. It's true that microbiology is fundamental and needs to be taught much more widely, and again, fundamentally. That microbial perspective is always with me, and I do my best to preach it to my students. Microbial centricity is one of my central philosophies. On the first day of class, my new Micronauts will select a glow-in-the-dark tag of an interesting microbe and dive deep into it for their weekend assignment, making things that are factual and creative. This example of the bacterial predator, Myxococcus xanthus, should give you some idea of the choices I provide. I've selected 29 candidates for the 19 students enrolled. We'll see which student selects which microbe as their microbial pal, and what they teach the course about their choice. One of the central concepts I present in my microbiology course is the cell wall that surrounds microbial cells. They are not simple and play many important roles in the life of a microbe. So I am very pleased to introduce my friend Dr. Dennis Claessen of the Institute for Biology at Leiden University to the podcast. Dennis will tell us about cell walls of bacteria, how some cells can do without them, and what costs and/or benefits result, and even how it can relate to antibiotic resistance. Welcome to the Quality Quorum today, Dennis, and thank you again for your courtesy when Jenny and I were in Leiden in 2022.
Thank you for having me, and I'm also looking back at a very great visit you had here with Jennifer, and fond memories of that.
It was nice. You took us to dinner.
Yes, together with Ariane, and we visited some nice musea here, the Boerhaven. I remember where you were allowed to touch one of the few Van Leeuwenhoek microscopes, right?
Mm-hmm. I was.
Yeah.
I think before we begin, Dennis, it would be a great idea for listeners and viewers, especially for people new to microbiology, to just define a few terms if we could. And I think that we need to know the difference between the cell wall, the cell membrane, and of course, a really swift, because it's connected, definition of L-forms.
Yeah, so I know quite a lot of people tend to call everything beyond the cell membrane the cell wall, which I think is a simple way to describe it. So the cell membrane is what circumvents the cytoplasm of a cell, is a lipid bilayer. But on top of that lipid bilayer, bacteria often produce a cell wall composed of different components. The primary component that all bacteria have is called peptidoglycan. It's basically a sugar polymer composed of N-acetylglucosamine and acetylmuramic acid that is forming basically in essence a large single molecule that circumvents the cell. Well, there are all kinds of variations to this theme. So some bacteria, diderm bacteria, they have a second membrane on top of their peptidoglycan layer. But in essence, it's everything outside what we call the cell membrane. So an L form basically is a bacterial cell that, at least for monoderm bacteria or gram-positive bacteria, lacks the majority or every peptidoglycan on its cell surface. So there, the cell membrane, we think, is what is exposed to the environment rather than the peptidoglycan layer. For diderm bacteria, it's not so clear yet how that actually looks, but for sure it lacks peptidoglycan, but we're not really sure yet if an L form of a diderm bacterium also lacks the outer membrane. Actually, we think it probably doesn't. So the cell wall, as we know it, is really a structure that is considered essential for bacteria. It's also a structure that defines basically how bacteria look like. So if we look at a variety of bacteria, which you can see here in this image, we see all these lovely shapes basically. And what we know is that these shapes are ultimately the result of synthesis of this outer surface structure called the cell wall. And basically the reason they look so different is because of the way they synthesize their cell wall, and also these shapes that all these different bacteria actually have has evolved to be beneficial in the environment where these microbes actually reside. And the fact that these cell walls are basically shape defining is that we can also isolate them actually intact, and it's really nice. You can basically isolate what we call a sacculus, which is basically an empty cell, which where you can only basically see the peptidoglycan cell wall layer, which is one of the key determinants of the cell shape and also of the cell wall. And sure enough, when I was in doing a postdoc in the UK in Jeff Arrington's lab, I got exposed to L forms. Before that, I actually hadn't known that much about it. But one of the postdocs there started working on Bacillus subtilis L forms, because there are so many at that time fundamental questions related to what these cells actually are. And one of the postdoc doing most of the work there, Mark Lever, he managed to obtain L forms of Bacillus subtilis, and that's actually when I got interested in these cells and thought, well, whenever I start, if that happens, my own group, I really want to do something with these L forms to see if I can solve some fundamental problems and do more on these on this exciting state of bacterial cells, really. So this is actually how it all started for me, more than a decade ago now.
Well, I think it's important for listeners and viewers to understand, and I've been guilty of oversimplifying things when it comes to the cell wall. And sometimes I talk about, how you can confuse the membrane with the cell wall, which is not the same thing. And the sacculus I love because it looks like a network, because it is. And that's our friend peptidoglycan. You have long strands of N-acetylmuramic acid and N-acetylglucosamine, a heteropolymer, and then linking those strands are short chains of amino acids, peptides. That's where peptidoglycan comes from. And sometimes you find D amino acids, which is the enantiomer of our standard L amino acids there. But what's interesting is that's a target for a lot of antibiotics. And I sometimes trivialize this because I tell, and you must forgive me for this, Dennis, I will trivialize the sacculose, the peptidoglycan, by saying it's Spanx for bacteria. Now, in the early days, we would call that pantyhose, and I'm not trying to be arch or strange with that, but it does help deal with like holding things in under challenging, in this case, osmotic conditions. So it's a wonderful thing to think about. A lot of people will consider a microbe and they'll say, well, this is like Tupperware, separating the inside from the outside, but it has to let things in and let things out, and all of it has to be carefully orchestrated. So the idea of living without the cell wall is really fascinating.
Yeah, I fully agree, of course. And the cell wall is indeed one of the key structures that provides protection to cells. And the bacteria I've been working a lot with are actually filamentous bacteria that grow at the tip. Well, you mentioned everything that goes in the cell and everything that goes out of the cell happens actually at the tip, while the tip also is the site where these bacteria actually grow. So it's puzzling to think what happens at the nanoscale at a tip of these bacterial cells, and while at the same time it has to be a permeable layer for molecules. At the same time, it's also the site where they grow, and frankly enough, also the weak side of the bacterial cell. And when my group started to work on these wall deficient bacteria, as I also like to call them, there's a whole variety of wall deficient cells. They actually escape from that tip as well. So and indeed often triggered by adverse growth conditions or antibiotics. So you already mentioned antibiotics, of course, perturb oftentimes cell wall synthesis. It's a great target for treating infections because, of course, human cells don't have a cell wall, certainly not like bacteria. So the antibiotics that we actually provide are great for killing bacterial cells in this case specifically. But at the same time, that also weakens the cells. And oftentimes what we see, at least in the lab, is that exposing these cells to antibiotics actually leads to the formation of these L forms or cell wall deficient bacteria. So it seems like a kind of ultimate stress response that allows the cells to survive at least for a certain amount of time without a wall. And I think that appreciation is now becoming more clear, although the first observations of this are almost already a century old, really, done in the 30s of the previous century, really. So.
And I like you calling them wall-less mutants because I think that's very accurate. The term L form invariably leads students to ask me, what does the L stand for?
Yeah, I get the same question when I talk about this topic. So the L actually refers to the Lister Institute in London, where Emmy Kleineberger Nobel actually worked on a bacterium in 1935, and showed that in one of her cultures she observed these what she called L forms, referring to the institute where she was doing her postdoc at that time. She initially actually thought, I reread some of these articles recently, that it was actually a symbiosis between the bacterium she was working on and this L form, which she wasn't really clear what it really was. And then later, Louis Dines actually in Boston discovered that it's actually the same organism, but a completely different form of the same bacterium, really. And nowadays we know that virtually all bacteria can, under particular conditions, make this switch to this L form state, as it's called.
I think it's interesting because we're so lucky nowadays in that we can actually determine the identity of something. In the 1930s, what were you were looking at the shape, maybe some biochemistry associated with it, and sometimes the nature of the cell wall, which is in essence what the Gram staining technique does. So once again, we're lucky to live in this period of time. And I tell students this quite often. You have to be grateful
True.
For how easily we can do things today.
Yeah, yet it's still really complicated to, to not to study them. I think in isolation. I think we've got pretty much the techniques in place. But if we start thinking, more in a community setting, right? So like in biofilms, multi-species biofilms, where these cells could also thrive, or even in a host environment, it's still very difficult actually to study what really these L forms mean in relation, for instance, to diseases. So I think we're just starting actually to begin to understand if there's any role, but it's really complicated because they look so pleomorphic, right? So you have spheres that look empty. You have they're all kinds of sizes, and if you imagine that in a host environment, it's really difficult to actually discriminate what is the L form or the bacterium and what is actually belonging to the host. So there's still some challenges there to win and to battle, basically.
And can I say that I want to applaud you, because I know how complicated filamentous bacteria are. And they are.
They are.
And if you're going to study an L form or a wall-less mutant, some people say, well, it should be in, and you mentioned Bacillus subtilis as an example, or E. coli for another. But you've done some of that, I know, I've seen it, but you mostly have the focus on filamentous bacteria, and you still find out fabulous stuff. Now, I think a good thing to do at this point for listeners and viewers is to tell them what the stressors would be on one of these wall-less mutants or L forms. What would they be experiencing differently than ones with walls?
Yeah, so these filamentous bacteria reside usually in soil environments, and soil environments are very heterogeneous. So whereas one patch may have low levels of salt, others may have higher levels of salt or osmolytes, and osmolytes are one of the key triggers that drive, at least in these filamentous bacteria, the formation of these wall deficient cells. So I think that's not really a surprise. So I think the levels of osmolytes or sugar or salts that you need to add is quite high. So this can be up to 0.5 molar, which is roughly the equivalent that is present in the sea, actually. So it's also not an amount of salt that is something that would never happen in real life for these bacterial cells. But at those levels, I think what we're really seeing is that at those concentrations, peptidoglycan synthesis really is going to be limited, or is going to be hammered, which leads to thinning of the cell wall. And we already talked about the osmotic pressure, for instance, inside cells is still very high, and actually so high that we think that is actually pushing these wall deficient cells out. Basically as a consequence of reduced ability to synthesize peptidoglycan. But at the same time, these wall deficient cells, because they are so fragile, they also need these high osmolyte environments, otherwise they would simply burst. So this is a bit of the trade-off between less wall capacity to synthesize cell wall, but at the same time providing the conditions for these cells to actually survive, and this also relates to the complexity, and I think I would say the ignorance that people have had towards these bacterial variants, because growing bacteria on plates with 0.5 molar of sucrose or salt is not something that you would do immediately. But so I think a lot of what we see is basically a better understanding what these cells need. But there's also the reason why a lot of this has been missed or is difficult to reproduce, because different labs use different media, you have standards.
When I was teaching introductory biology, one of the things that really grabbed students, we would take red blood cells, and no, they weren't from the students, and you would put them in distilled water, which has no solutes out there. And so water would rush in, because that's what osmosis is, and the cells. All right? And you could watch it happen. It's that fast. And if you put them in higher levels of salt, then the red blood cells are pushing water out, again, to balance total solutes, and they get all wrinkly and constricted looking. So that business of protecting cells against changes in osmotic pressure is critical, especially because the natural environment of bacteria is not a petri dish or a culture tube.
Yeah, true. Yeah, no, that's true. And yeah, so actually, as you started this question, so with streptomyces, it was actually a serendipity that we found this ability to make these wall deficient cells. It wasn't really that we were desperately looking how to make them, but it was. So these streptomyces, they're not the most easy bacteria, these filamentous bacteria, to work with, and they use very different media than I was used to when I worked in Jeff Harrington's lab on bee settlers. So actually, the funny thing is, for some cultivations, you actually use naturally high osmolyte environments, and that relates to how these streptomyces grow. They grow very filamentous. They make these complex multicellular networks. They're interesting bacteria for industry because they produce a lot of antibiotics, but the way they grow is not so favorable for industry. So one of the solutions industry, or at least came up with, is use high levels of sucrose or osmolytes, because they grow more dispersed and they produce more important molecules that industry got interested in. So actually, when the only thing we needed to change to get these strains to make these wall deficient cells was to reduce the shear, so lowering the stirring speed. And actually, if you grow a wild type streptomycete, or at least a good number of these strains, at a lower speed, you get cultures full of these wall deficient cells naturally, because they are actually not so happy, but under normal conditions actually get killed or get by the shear stress that you impose upon them. So people have just missed it, because people just do what the protocol says, right?
Yes. And this is wonderful. This is a great thing for students to hear. I love the business of the selection in favor of forming what in the old days we called spheroplasts, cell wall-less microbes, because the ones with walls were more susceptible to shear forces in a fermenter, because that wouldn't have occurred to me. In retrospect, it does. But this is the thing I tell students. It doesn't matter what expensive piece of equipment you have, millions of dollars worth of things, doesn't compare to your observational skills.
Yeah.
And this...
every now and then forgetting something or making a mistake actually can help a lot. So this also relates to how Jeff actually discovered the L forms. It was also kind of a mistake from a swapped medium that students used with bacillus, which is normally used to make protoplasts, and which has high sucrose media. And a student forgot actually that, and that was the basis of the discovery of how to make these L forms.
Well, it's interesting too, because if you think, when you hear about this business of stabilizing a spheroplast, an L form, a wall-less mutant with high levels of sucrose. So the idea is, for most bacteria, they can't take up sucrose. There are some exceptions.
Yeah.
But so that means that you have a lot of solutes outside the cell. And that means that that's going to be better than a situation where the bare bacterial membrane is exposed to much less of an osmoticum, say in culture media. So what a wonderful thing to do. I am familiar with people that will use enzymes that attack peptidoglycan, and that can lead to the formation of spheroplasts, but that's not what you're talking about. You're talking about an advantage that occurs under those conditions.
Yeah, so it's really under these conditions that, these high level of osmolytes that, basically the cell can no longer grow happily, and I think as a consequence starts producing these kind of cells, which we now know also provide benefits to all kinds of other stressors. For instance, one of my favorite things that I've recently started working on are bacteriophages, actually. So um, bacteriophages recognize epitopes in the cell wall, and sure enough, if you make these kind of L forms, we see that many strains no longer are susceptible to making to become infected by the phage. So there are definitely conditions imaginable where these wall deficient cells are very, very useful for the bacterium.
May I ask a question about that?
Sure.
So the idea for listeners and viewers is that bacteria have receptors on their surfaces, proteins on their surfaces, and that's what the bacteriophages can recognize. And there are some exceptions to that rules, of course. Just bear with me for a second. However, if you're an L form, if you're a Wallace variant, whatever we're going to call them, are those proteins no longer there?
Well, it depends where the receptors are, really, right? So you also
Right.
have receptors in the membrane. Yes,
Mm-hmm.
then of course the phage will still be able to recognize those and start the infection process. But there are also teichoic acids or LPS structures that are used as
Mm-hmm.
receptor. Um, yeah, and without those, phages will no longer be able to recognize such cells, and will not be able to infect them. In fact, it's a nice. There was a nice follow-up paper from my student Veronique Ongenay that I supervised together with Ariane Briegel, who has also been on your show. So she discovered that this behavior was happening in actually quite a number of bacteria. So if you infect the population, you always get survivors that seem to rely on the formation of these wall deficient cells to survive. And then there was a follow-up paper from Martin Leusner's lab in Switzerland that showed that actually one of the enzymes, an endolysin that is used in the late stages of the infection process that usually degrades the peptidoglycan, helps to provide communal protection to the whole community. So the endolysin basically also doesn't only act on the cells that have been infected, but also strips off the cell wall of neighboring cells that had not been infected yet. And by doing so, those cells that lost their cell wall but had not been infected were actually protected against phage predation. So I think that's a fantastic example how this basically works, or how this could work, and how this may have also worked billions of years ago already when when life started, really. But nowadays modern cells still may be able to use.
It's interesting. Bill Metcalf, a friend of mine from long ago, who's a professor at the University of Illinois at Urbana-Champaign, used to tell me that when you're doing bacterial genetics, a phenotypic screen that you're using, that's not the bacterium's agenda, that's yours. And we say, oh, they're slippery because they're not following the phenotypic screen that we expect them to. But you see, we're wrong. They're living their lives. So there's a couple of things here that you said that I think are really important. And the first is microbes that have interactions with other microbes around them, even if they're SIBs, as you just mentioned. So this kind of community structure that we're used to when we talk about animals and plants also hold true in the microbial world, right?
Yeah.
The other thing is that we get very narrow in our knowledge these days. So I look at bacteriophage lambda of E. coli. It recognizes the maltose receptor. And I know, because I've done it, if you find mutants that can't take up maltose, a large proportion of them are resistant to phage lambda infection. That makes sense. But there are other types of bacteriophages, as you mentioned, like phage P1, that uses LPS to get in, and that's going to be common to everything. So once again, having some broad knowledge of the depth and breadth of the microbial world is fundamental.
Yeah, and I think I'm in that sense a bit biased also with the model bacteria that we use. So these Streptomyces are
Yeah.
really multicellular bacteria with differentiation happening, like, well, at least in some ways similar to what happens in higher organism or eukaryotic cells, right? Specialization of filamentous structures to terminal differentiation, and
Hmm.
so yeah, it's that pair of glasses, I think you would call it, that
Yeah.
helps, of course, to disentangle these processes that happen, between bacteria and phages, or between bacteria amongst each other.
But I want to, defend what's been done, because sometimes people hear it when I get speechifying on this subject. Uh, I'll say that single colony isolation and growth in culture tubes doesn't reflect nature, and it's true. Neither of those things do. And it's also true that bacteria evolve under those conditions, because they do. But how else could we have found things out to begin with? So it's easy for me to say that we depend on model systems, because we do. And I call that coli centricity. And but at the same time, I don't want to disrespect the people that did all that early work, because it was really hard work to do. The problem is we have to be open to how diverse the microbial world is.
Yeah, fully agree. It's There's so much to discover in this respect, and I'm also really happy that we see much more different bacteria being studied in the past decades, I would say. I think we learned a lot from E. coli. We will in the future learn
Mhm.
still a lot from E. coli, but we will also learn a lot from other systems that are now being developed, and that gives us this complete picture in the long run, how bacteria work, or at least a little grasp of how they work, because they're immense complex, and certainly in association with other bacteria, with the tricks they can do. And yeah, no, it's fascinating. It's fascinating to be a microbiologist, I think.
And I love the idea that you were able to show in multiple publications how bacteria that are wall-less can be resistant to bacteriophage infection, which is, depending again on the bacteriophage, which is just tremendous and fits so well into things that we're talking about. I do wonder why it is we can't, or maybe perhaps we can, make mutations in the pathway that make peptidoglycan. And can those bacteria then form the wall-less form normally?
So we've been pioneering a bit in this avenue as well. Jeff Arrington has also done this. So once they are in this L-form, a wall deficient state, you can actually get rid of proteins involved in peptidoglycan synthesis. So in one of Jeff's papers, they deleted a large region around MRC, which is one of the essential genes that is involved in synthesis of peptidoglycan. And if you do so, you basically block the ability to rebuild peptidoglycan. So
Mm-hmm.
these bacteria are kind of indefinitely locked in this wall deficient state. So a lot of L forms, if you make them in the lab by exposing them to conditions that promote their formation, you will end up with L forms that will always be able to go back to this walled state. And I think that is actually one of the most fascinating things, right? That you have bacteria that can readily switch between these two states. So that relates, for instance, to also diseases. So my lab is currently looking in Mycobacterium tuberculosis, which is also an actinobacterium that, of course, kills more than 1 million people each year. And what we have seen is that this bacterium is also quite happy in making these wall deficient cells under inducing conditions that we impose upon them in the lab. But they also have the ability to go back, and so it basically offers the bacterium a kind of shift in shape and also in physiological state that allows it, for instance, to resist antibiotics, right? So if there are
Mm-hmm.
antibiotics in the culture, it can kind of needless switch into this wall deficient state where the antibiotic won't be able to kill the bacterium anymore because the bacterium happily survives without a wall. But if the antibiotic gets the therapy is ending, they can actually go back to this wall state and potentially make you sick again. Of course, we don't know if this is one of the mechanisms by which Mycobacterium in this way persists in the host. This is something very difficult to study, but it's also not unlikely that this may be happening. And at least for E. coli urinary tract infections, one of the big problems that a lot of people suffer from, they actually identified in urine samples E. coli cells that had this ability, right? They could switch from one to the other state. So fosfomycin treatment that is usually offered to these patients leads to the formation of wall deficient cells. After the treatment, these wall deficient cells actually revert to the wall state, and we think that could be a main reason why these people get reinfected after treatment. And the appreciation for this is, I think, going to be future change important for the future to change how we prescribe also antibiotics. And this is, I think it's pretty fascinating.
It's interesting. We've talked about antibiotics and antibiotic resistance quite a bit on this podcast, and we should. And what occurs to me is that we tend to, again, I sound flippant when I say this, as humans, we're very narcissistic and we're mostly concerned about how things interact with us. I'm not putting that down. The early days of microbiology came up with solutions to a lot of human and animal diseases. That's really important. But we tend to, and I'm not going to say anthropomorphize, but we tend to think we know what the strategy is, why a bacterium does something. But the fact of the matter is they're just responding to their environment. And they're endlessly protean, endlessly changeable. And you are an environment in which they're living. So with the example of Mycobacterium tuberculosis, I mean, it's adapting to live in you. And this is one strategy to avoid, as you've mentioned, antibiotic treatment. And given how important, and folks, listeners and viewers, if you don't know, and I'll put some links in the show notes on this, if you don't know how important tuberculosis is, historically and currently, time to do some more reading. Because there are all these different types of Mycobacterium tuberculosis that are highly resistant to antibiotics. So we got a problem. So understanding this process is actually involved with health, Dennis, in a way that you know and I know, but I want everyone else to know too.
Yeah. No, I think most bacteria, are not there to kill us or to make us sick. Mm-hmm. It's just a dysfunctioning of the microbiome. They're living with millions of others that gets disturbed. And I think they just like to survive like we do. And one of the ways they could do this is to try to overcome the antibiotic challenge they're exposed to. And one way is potentially by making these wall deficient cells. So fully on the same page here with you.
It's so interesting to me because I'll have students make antibiotic resistant mutants of the bacteria that we study trivially. It's easy to do and calculate the mutational frequency and all the rest. And then I tell them to sit down and really think about what they've done. Look at streptomycin resistance, for example. And that helps me get them to think about what a ribosome is composed of and the genes that encode those portions of a ribosome and how a single nucleotide change can cause an amino acid change, a tiny change in the shape of that portion of a ribosome that makes it resistant to, say, streptomycin. And it's really interesting.
Yeah. And frightening every now and then, right? Mm-hmm. That it can happen so quickly.
So this has been very exciting stuff. And I like the idea that you've talked about several projects. And I know there are others that you'd like to talk about a little bit, I think.
So one of the things that has fascinated me with these wall deficient cells, is to one of the theories actually that Jeff Arrington already proposed. You see, I mentioned Jeff Arrington a lot. I'm one of his postdocs that started his own lab. But it's the link to early life. I think that is for me really really fascinating. So, um, which also links to one of your earlier questions. So if you think about these L forms, they're happily living without a cell wall. But we also know, as I also explained, you can actually delete genes related to cell wall synthesis, but also cell division. As cell division in bacteria is pretty conserved, I would say, and one of the proteins that is almost invariably involved in this process is FTSZ or FTSZ, as we say it here. And this forms like a scaffolding protein for all other proteins to come together to mediate cell division process. And I think in one of the early papers from the Arrington lab, they showed that these L forms don't need FTSZ anymore, and that was the start of this whole idea that these kind of L forms, also follow-up studies showing how they actually proliferate without cell division machinery. Maybe a great example how early life forms, so we're talking about bacterial cells before invention of the cell wall, could have lived. And I think that is super fascinating. And if I tell that to students, they're always like opening their mouth like, oh, wow, are we looking now at ancient bacteria? And then I say, no, but we may look at a process that is already billions of years old, actually. So we're still looking at a modern bacteria, but maybe behaving a bit like how early life forms have behaved. And I think that is for me pretty fascinating to think about, and that you actually only need 1 or 2 point mutations to get to that state, really. That maybe billions of years, they have led to bacteria that have all kinds of shapes, make these beautiful peptidoglycan layers and other more complex cell walls, but that they can go back to this kind of primitive mode of growth. I really like that connection that has been proposed.
It kind of smells to me a little bit like, and there's nothing new about it, but it's kind of bounded onto the page, so to speak, in a lot of journals, extracellular vesicles, because I remember them in a very trivial way back in graduate school. But now it turns out that they're everywhere, carrying all kinds of things. And that might be an echo of that early form of life that you're discussing about.
Yeah, I think so. The biology of these extracellular vesicles, gets a lot of attention in bacteria. Of course, in the eukaryotes, it's always been a hot topic. But I think having something in hand to study, what happened maybe billions of years ago, I think is it's really really interesting, and of course also ties in with the synthetic biology approaches that Craig Venter and his team has been pioneering, right, to create minimal life forms. And in one of their papers where they created Cynthia, I think it was version 3.0. Actually, that's an L form. So which only required 470 genes or so. So I think, I love these kind of things.
It's always funny. Students will ask me questions like, they'll say, is the evolution of life, did it follow this path or that path? And I'll say yes. I'm being silly, but I also am being accurate, because we don't have a time machine to go back. We make guesses, but we can't know for sure until we start making life forms, for lack of a better term. And I know that when I say that, a purist would say, well, no, you're taking the end products of evolution, just shuffling some genes up and finding the minimal number. Well, sure. But it gives us some insights into what's necessary for life. Most people would think that you need something like a cell wall, simply because of the prevalence of cell walls among a lot of microbial life forms. But as we're learning, that's not always so. And the conditions in which life evolved could have been quite different.
Yeah, true. No, so of course I agree with these arguments that we're not, of course, not looking at these early life forms, but it just tickles your mind to also think how could it have happened, right? So
Hmm.
one of the things I'm really curious doing is, so if we have these wall deficient cells or these L forms which cannot build wall, how are they going to evolve a cell wall? This is one of the things I really would love to study in the future. So you take this minimal cell, or you take an L form, you strip away everything that we know about peptidoglycan synthesis. How do they reinvent a cell wall, for instance? And it may not be the answer to how it evolved, but it gives you an answer how it could evolve, like you
Hmm.
say, right? And maybe I can spoil a cool hypothesis that I have. So if that's okay. so I,
Sure.
so I so if you think about a cell wall, it's really a sugar polymer, right? And
Hmm.
um, of course, sugar is also food for a bacterium. So one of the ideas I like to think about is that maybe a cell wall actually emerged initially as a storage for food. So if you are able to couple food to yourself, it gives you, first of all, some primitive protection for fluctuating environmental conditions, not as, of course, as robust as a peptidoglycan layer would be, but as say you make little sugar chains connected to yourself, you do have some protection. But secondly, you're able to move, or you're able to go away and take your food with you, right? So this is something I find quite interesting to see if we can find ways that would support this kind of idea, that maybe um, life or cell wall, basically biogenesis, started as basically a food storage reservoir. And these kind of things I think are quite interesting to think about. And
Oh, I think that's a really fabulous idea. I've spent many years studying bacteria that are very, very slimy on plates. They're making what's called an extracellular polysaccharide. And the fast-growing rhizobia that I did my PhD work in, part of that extracellular polysaccharide is a signal to its partner to form the symbiosis. But it's also a fill in the blank for your favorite expletive to work with, because it covers the cell up, it's hard to break the cells, etc. As you say, if they are coating themselves with a capsule, it serves some of the purposes of a cell wall,
yeah,
plus a mobile food source. I like that idea a lot. And, it'd be easy to test that kind of thing out in terms of survivability or evolvability with, say, I'm making this up, a bacterium that makes EPS and one that doesn't due to a single mutation, and see how they compare fitness wise
yeah,
under stressful conditions.
yeah,
Because it's well known, it helps protect against dehydration, like Azotobacter does it as well.
yeah.
So I think your idea is fabulous.
I mean,
My opinion doesn't matter, but you know what I mean. I think it's a fabulous idea.
yeah, yeah, and it's also in line, of course, that also modern bacteria recycle about 50% of the peptidoglycan every um,
Hmm.
every doubling, basically. So they basically continuously eat their own peptidoglycan
Hmm.
to make new ones. So yeah, um, first get some funding to do this kind of work.
Well, there's precedent for it, because a lot of people, I'm including myself with this. I believe that horizontal gene transfer is really an aberration of obtaining food. And it's what Rosie Redfield used to call the genes for breakfast hypothesis. And the whole idea is, in all the cases that I'm aware of, other than, well, and sometimes with plasmid transfer, you get a single strand and the other one gets broken down and recycled, because DNA is a polysaccharide.
Yeah, yeah, yeah.
So,
Starvation is the key trigger to get competence in bacteria, right? Or at least
mhm.
in a good number. So yeah,
But again, like I said earlier, if we have something we think we understand, we put our point of view on it, we're not open to new ideas. And that's why having the time to sit and chat with colleagues about it and kind of brainstorm is important. As we chase grants or writing papers, whatever, it's hard to do that, or teaching, very hard to do that. But honestly, you can see there's lots of ways to bring ideas together.
I think in the past we also discussed another cool idea, right, about Vibrio.
Yep.
So these bacteria eating bacteria really, where the wall deficient cells could be maybe, well, not so eatable for them because they may not be able to, yeah, to recognize them or to eat them. But this has never been done yet.
And, and I know it hasn't, and I would love if I found E. coli or Pseudomonas that, I mean, which would be good to try. But I wanted to say that I had been asked this so many years ago. Couldn't I just treat E. coli with lysozyme and then add high sucrose on it to preserve them as spheroplasts? And that's one way to do it, sure. But I did want to add one thing, and I want listeners and viewers to understand. When you change something fundamentally about an organism, and it was used to having its other way of living, it's often resistant to change. So there is a cytoskeletal element in bacteria, MREB, that controls the difference between a sphere or a coccus and a rod as a bacillus. And so E. coli with that rod shape, an MREB mutant looks like this. And you think, aha, it's all it's needed. But if you look at that mutant carefully, it has a number of different mutations elsewhere that help stabilize it. And so one of the problems we have when we knock out a gene is that we're not really looking at the effects of that change on other genes. It's not as easy to look at, and we miss stuff that way.
Yeah, they quickly pick up compensatory mutations to make them feel better. Yeah, that's also what happens with L forms a lot. Actually, they're not the same if you culture them for a few generations. They're not necessarily what you think you had in the beginning.
Well, the advantage today is you can actually find out what genes changed from your original strain and your others. And that was something that I couldn't believe that I could do that in my own lab for about $80 a throw.
Yeah, crazy.
That's pretty amazing. Yeah. And you know what? In a few more years, I'll be able to do it on my phone.
Yeah.
From the App Store, right?
True.
So as we start to wind down a little bit, and I have just loved this discussion so much, and I hope I haven't gotten carried away with my own views of the microbial world, but I wanted to ask you, because I have so many students that think there's only one way to become a microbial scientist, could you do the following, please? Talk a little bit about your path to becoming a microbial scientist, and then if there were 3 things you would like folks to remember from this discussion today, what they'd be.
Okay, my path to becoming a microbiologist started actually when I started studying biology in Groningen. And there we actually had a lot of classes in the beginning about plants and drawing plants. And at that time I thought, well, I'm not sure if this is really what I would like to do. Until we got a series of lectures on fungi given by what would later become my promoter, Han Weuste. He's a very inspirational microbiologist, also happened to was born actually in the same city as I do. Well, the Netherlands is not as big, but it was quite a coincidence that actually his father taught me in primary school, really. So his name is also not very common here. So we immediately had a click, and he was just very, very inspirational. And he was so inspirational that I said, well, I really want to try to get a PhD under your supervision. Which he said, I'm going to try that. Of course, he also needed money for that. But meanwhile, he got a new position elsewhere on a different topic. So he was working on filamentous fungi, but had moved to a different group and started working on filamentous bacteria, streptomycetes. So actually, I said, I don't mind. I just want to work with you. So the topic at that time didn't really matter so much to me. So the fact that I, in the end, still nowadays work with streptomycetes was because he worked with them in the past. So so in that way, the path was basically driven by an inspirational mentor. I think, of course, a topic you should really like, but I think in the end, it's the combination of a topic and a excellent supervisor, somebody that's there when when the world goes a bit black and you think everything else, everything fails. That is what you really need, I think, and that's what he could give. That's in the end what made me go into microbiology. Of course, then I did a postdoc, etc., in Jeff's lab, which I already discussed extensively. That's what brought me basically here. And then your second question was 3 things that people should remember. Yeah, so maybe the first one is actually related to the last part, right? So try to find somebody that's inspirational. And I'm not sure if your students watch this, but I know you, and you are very inspirational. So I know you've given quite a few people or led to in the end PhD. So I think that is really one thing to keep in mind, regardless of the topic. It can also, of course, be any other topic, but for sure, find somebody that you can connect with, and that can help you shape your path. And then I think the other 2 could be a bit related to, of course, the content we discussed. So try to make every now and then an error, but keep looking what you see then, right? So it's okay to make mistakes, but learn from those, or find new things to discover, because there's an awful lot to discover. And the other thing would be, I think, to really take these L forms more serious. I think people have neglected it. I think it's slightly changing now as more groups are working on it. We better understand it, but we're still not there yet, right? So I know the medical field is quite skeptical still about these shapes, and rightfully so, because we have no direct clue that an L form can make you sick or cannot make you sick. But I think we are now in a time where we can really solve these questions. So go hunt them. Go hunt for the answers related to that. So I think those were the 3 things I would leave with the audience.
I think this has been a wonderful session, Dennis. And I really want to thank you for taking the time to chat with us. I learned a lot, and I hope listeners and viewers did too, because there's a lot here. And I'm always surprised the number of times that I'll think about a change in one aspect of an organism, and I'll go, but it turns out this is so central in so many areas. And it's a great example of how a simple observation, a path that you take, can create a whole career.
Thank you very much. It was a joy being on your podcast.
I wish you and yours the very best, and I hope to see you again in the future.
We'll definitely make that happen. Thank you.
Very welcome. This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, with, as always, epic links, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Dennis Claessen can be found in the Institute for Biology at Leiden University in the Netherlands. Many, many thanks, as always, to David Renata for superb editing and Reber Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our Quality Quorum for today. See you next time on Matters Microbial.